want to introduce our three speakers. We're really lucky to have this be a tag team presentation. So, leading us off is going to be Dave Clapp. So, some background on Dave for those of you that I think many of you probably know Dave and doesn't need an introduction given his long career with Michigan DNR. Dave got his bachelor's in science at University of Michigan and then his master's in science with Jim Diana, also at U of M, in the School of Natural Resources and the Environment, I think it was called then. Dave, is that right?
Just
Before,
just SNR back then.
oh, before the E.
Yeah,
Okay,
yep,
there
yep.
you go. So Dave then had a 28-year career up at the Charlevoix Fisheries Research Station for Michigan DNR, retired in 2023, and now has what he describes as a perfect job with the Nature Conservancy as a conservation biologist. Um, I think next in our talk will be Matt Herbert. Again, many of you know Matt, I'm sure. Matt got his bachelor's in science from Iowa State University in fisheries and wildlife biology, and then got his master's at Texas A&M University in wildlife and fishery sciences. Matt is the senior conservation scientist with the Nature Conservancy and has been with TNC since 2007. And then they're going to pass the baton to Taylor Skiles. Taylor is currently a technician with Michigan Department of Natural Resources. She has her bachelor's at the University of Wyoming. She was just telling me before the early on that she worked on a project there with Willie Fetzer, and that was potentially part of what brought her from originally Nebraska to Wyoming and to northern Michigan. And we're really excited. I've been working with Taylor on the swim project and excited to learn that she's going to be starting a master's program in the fall with Scott Colborn and Dale Hondorp at Michigan State University. So again, the three of them are going to tag team. You've heard their introductions. They said the talk is about 40 minutes or so. And we hope that all of you will stay on after the talk to ask questions and we can have a friendly discussion about their work, which is Northern Lake Michigan reef evaluation and prioritization. So take it away, Dave, and thanks.
Yeah, great. Thanks, Bo and Renee, and thanks everybody. We're happy to be here to talk to you today. We're going to talk about a study that we conducted in 2022 to 2024, and just had a final report come out last year. But we want to provide some early results, and then hopefully talk a little bit about how what we found might be translated into some habitat management recommendations. So, given today's the webinar topic, we're going to focus hopefully a little bit more on whitefish and herring. Um, and we got tons of partners and co-authors and collaborators here, but I want to make sure to acknowledge Patrick Hanshin. He kind of really did a ton of work wrapping this up at the end and kind of getting us to the finish line. So acknowledge Patrick here, and also then throughout the talk we'll be acknowledging some of the other folks too. But with that, we want to talk about this project, and as fisheries managers, our job is to protect and restore fish stocks, and we have several tools or levers that we can use: stocking and harvest regulations. But habitat management and habitat protection is really key. And an example of that is the Great Lakes Restoration Initiative focus area for the habitat protection focus area. Projects funded in this area throughout the Great Lakes indicate the importance of this to fish populations and a healthy environment in general. So I think everybody here's familiar with Great Lakes reefs. We give like more of a public presentation. We have to talk about coral reefs and colorful fish and things, but we're talking about these rocky areas out in the Great Lakes. Really important habitat along with rivers. They're really an engine for fish production. And if we have, they provide food, they provide areas for eggs to be protected and young fish to be protected in the interstitial areas on these reefs. So really important habitats. If we have healthy functioning reefs, we have healthy fish populations and health and a healthy ecosystem. So um, feel like they're really an important part of the lakes here. Um, the importance of reefs has been recognized in kind of Great Lakes policies and uh, uh, Lake Committee and Great Lakes Fish Commission documents over the years. And I'll focus a little bit. I'm familiar more with Lake Michigan. So in Lake Michigan, we had early on, this was probably late 90s, 2000, developed the Lake Michigan environmental objectives that recognized reefs as important. Ed Rutherford was really uh, kind of spearheaded that effort. More recently, all the lakes have developed environmental principles. Um, and then from that environmental principles exercise, priority habitat projects or habitat priorities were identified. And that was really the Lake Michigan environmental priorities, the first round of that, the protection, assessment, and restoration of reefs in northern Lake Michigan was identified as part of that effort. And then that really was uh, kind of drove the project that we're going to talk about today. That was the impetus for our project. So, um, but through the years, the importance of these habitats has really been recognized. Given their importance, we really don't know a ton about reefs kind of comprehensively across the Great Lakes. Several reefs have been studied kind of in depth. Um, uh, think about in our office at Charlevoix when I was working with the DNR, we did work on a few reefs in Lake Michigan and Lake Huron, um, and really got to know a lot about those specific reefs. But we don't have a sort of comprehensive understanding across the Great Lakes of reefs. The Goodyear Atlas shown on the left here, that's kind of widely cited as a source for information about reefs. It's great, but it really doesn't provide a lot of detail. It kind of provides general location information and some information about what species might use those reefs, but not the detail that we're going to talk to you about today. And other resources similarly don't really have the coverage and or the detail that we're going to be talking about today. So with that, we're talking about our project goals here. We want to advance our understanding about the distribution and conditions of Michigan reefs, Lake Michigan reefs. This includes things like physical characteristics, the substrate, just the size. Some of these reefs, we don't really even know what the extent of the reef was before our evaluation. So we're going to talk about physical characteristics, aquatic communities. Um, we did look at invasive species, but as I said, we're going to try to focus today on some of the information we have about whitefish and herring. Um, some of the characteristics that might contribute to native fish production. So what's important for producing fish off these reefs, excuse me, and then hopefully then translate some of that information into management actions. And I'm going to come back to that at the end and talk a little bit about how we're starting to do that. So with that, I think Matt's going to take over now and get into some of the specifics of what we found.
Thanks, Dave. All right, so the first thing that we did was to map reefs across Michigan waters of the main basin of Lake Michigan. We used the reef layer available through GLAF, the Great Lakes Aquatic Habitat Framework, as well as reefs named in Goodyear and reefs named on navigation charts to identify locations of reefs. We then created rough maps of what looked like the boundary of each reef based on navigation charts and LiDAR data where it was available. We also use LiDAR and navigation charts to identify additional reefs that weren't named in those prior efforts. And then once these were mapped, we divided uh, we divided them into broad regions, so you can see kind of a North Shore region, Grand Traverse, Little Traverse Bay region, and then more southern Lake Michigan reefs where there aren't as high a density aren't as high of a density of reefs. Um, uh, Reefs were selected for assessment to include a mix of geographies and landscape settings. So for instance, we wanted like some representation across those three geographies, and we wanted the kind of the conditions of those reefs. So for instance, we wanted a variety of exposures. So we wanted some reefs that were in bays. We wanted some reefs that were highly exposed to wave action. So we wanted a variety of conditions so that we kind of can compare across those sorts of things. Um, we also selected reefs that were more accessible, so we focused on nearshore reefs, and we chose groups of reefs so that we could focus work in an area where we could potentially work across multiple reefs within a day. Uh, the reefs that we worked on are in red here. So all the black ones are just reefs that we mapped that we did not conduct assessment on them, and anything red are reefs that we assessed. We conducted assessments on 24 reefs. There were a handful of other reefs that we visited or mapped. Some of those we collected some data, but we didn't collect a critical mass of data. So there were 24 reefs that we collected, kind of that critical mass of information. Next slide. All right, so once reefs were selected, our colleague Gus Annis led us through a process to map each reef in greater detail. So we had these kind of rough general maps. We wanted much better detailed maps of each reef. So we used bathymetric LiDAR data where it was available. We also used multi-beam data collected by Pete Esselman with USGS, who was kind of working in tandem with this project, collecting data on many of the same reefs that we were working on. We used the benthic terrain modeler in ArcGIS to identify core reef features by comparing the elevations of an inner radius of 15 meters with the elevations of an outer radius of 150 meters. So basically, we're trying to use this process to identify areas that are significantly raised above the surrounding lake bed. Areas where elevations were more than one standard deviation above the elevation of that outer radius, we identified that as core reef habitat. Then the core reef features that we identified were then buffered with a 200-meter buffer to define the greater reef complex. So if we go to the next slide, we can see what a couple of these reefs look like. So on the left is Good Harbor Reef, and on the right is Lighthouse Point Reef. And so the areas in color are those features, those core reef habitats that were significantly elevated above the surrounding area. And then the gray areas are basically define the whole complex. And again, those were just drawn by buffering the core habitats. And so this really helped us to drive where we were going to be doing sampling. For most things that we did, we conducted sampling both in the core habitat and in other areas, in those other areas within the reef complex. Next slide. So for each reef, we assembled information on the geomorphology of each reef and landscape characteristics of each reef. So on this list, the first four are related to the geomorphology, and the rest are all landscape characteristics that we calculated, basically, mostly using data available through GLAF. Later, Dave is going to talk at least a little bit about some of the analysis that we're working on now to identify patterns across all of these reefs. And that's where a lot of these data are being used, especially those landscape characteristics. Next slide. We collected data on substrate, embeddedness, interstitial quality, and some other habitat features on these reefs using drop cameras and also using diver measurements. Camera images were collected in both core and those buffer areas on both core reef habitat and those buffer areas. Diver surveys were quadrat sampling conducted at random locations across a transect. And we used CMAX to classify substrates. So I think CMAX is a common tool that I think NOAA and USGS, Fish and Wildlife Service, a lot of other folks are using CMAX. So we stuck with that CMAX classification for our substrate classification. Next slide. So these are drop camera substrate results. Uh, each reef is along the x-axis, and the y-axis is the proportional dominance of rocky substrates. You can see that for most substrates, or for most reefs, rocky substrates were dominant in at least half the locations, with cobble usually being the dominant substrate. Uh, the the Along that x-axis, the reefs are ordered along that axis from south to north. So starting with those two reefs in the very southern part of the lake, moving up to the Leland area, which is outside of Grand Traverse Bay, then into Grand Traverse Bay. You can see the labels here at the top here. So then we've got the west arm of Grand Traverse Bay, then the east arm of Grand Traverse Bay, then the Little Traverse Bay area, and then along the North Shore. And you can see, you know, something that we'll see in subsequent slides too. The west arm of Grand Traverse Bay tended to have a lot more habitat that wasn't dominated by rocky substrates, whereas the east arm of Grand Traverse Bay had a lot more dominance with rocky substrates. We know, the west arm, for example, has is a lot more sheltered. The east arm tends to get those northwest winds, tends to get hammered by those northwest winds. So that's something that we noticed a lot. You can also see those North Shore reefs. Some of those reefs also had pretty low rocky dominance. Next slide. We developed an interstitial index based on the dominant rock type and the depth of the stacked dominant rock type. So a combination of coarser substrate, if it was cobble or boulder, and if that substrate was stacked several layers deep, would result in a higher interstitial index score. And again, the x-axis is all the reefs going from south to north, and the y-axis is the proportion. So anything with low substrate we pulled out of this. This is just the proportion of drop camera locations that had either medium to high interstitial scores. So the interstitial score of 2 to 4 is kind of medium. Anything above 4 is you're starting to get really high interstitial scores. And again, you can see the west arm of Grand Traverse Bay had pretty low interstitial scores, as did some of the North Shore sites as well. And the east arm of Grand Traverse Bay tended to have the best interstitial. If you notice, especially those higher scores, those greater than 4 scores, you could see that both the east arm of Grand Traverse Bay and that Little Traverse Bay region had a lot of locations with really good scores. The many of you probably are familiar. There's been a lot of work done on Elk Rapids reefs, and so many of you are probably familiar with those because they get talked about a lot. And as you can see, that reef kind of stands out as having a lot of high quality interstitial. But you can see that is not common among other Lake Michigan reefs. It does really kind of stand out. Next slide. We also characterized the proportion of rocky habitat that was embedded. So embedded is basically where either sand or silt or even in our case, a lot of times it's mussels and shell hash has filled in all the spaces around the rocks. So you basically have a continuous surface of rocks. You can still see the rocks, but all the spaces in between are kind of filled in with either silt or sediment or with mussel shells. And so you basically have no space between the rocks. And we know, with fish, that's not a good thing. So a high embeddedness basically means there's not very many spaces for eggs to kind of work their way into the rocks. And again, you could see that west arm of Grand Traverse Bay had really high embeddedness, as well as a lot of the sites along the North Shore. Whereas the east arm of Grand Traverse Bay and a few other locations had really low scores. And again, Elk Rapids and some of those other sites adjacent to it have really stand out. Next slide. So just a few like takeaways from the physical habitat. I didn't go into the depth, but our mean depth was around 6 meters. That gives you a sense of like of what kind of reefs, what kind of nearshore reefs we were working in. Cobble was the dominant substrate on 19 out of 24 reef surveyed. Uh, we had high interstitial spacing was not very common. It was pretty rare, especially the medium interstitial was not so uncommon, but those high scores were quite uncommon. And the interstitial did vary a lot among reefs. Those more sheltered reefs in the west arm of Grand Traverse Bay did tend to have more shell hash and sediment buildup, creating those embedded conditions. And then I also didn't talk about the cladophora, but there is a lot of cladophora and aquatic vegetation is quite common, and it often is related to those habitats that are highly embedded. And then with that, I think Taylor's taken over from here.
Yeah, thank you, Matt. So I'm going to get us started on our third objective, which was to document the presence of invasive species on these Lake Michigan reefs. So we were interested in measuring the biotic factors that have the potential to negatively impact egg and larval fish survival on our reefs. So to do this, we used a combination of on-site and in-water surveys to quantify documented egg predators like round goby and rusty crayfish. But we were also interested in quantifying the presence of drusenid mussels because of their potential for negative impact on native fish due to fouling and resource competition. Not only did we seek to quantify the presence of these AIS species, but we were interested in connecting their presence to the physical characteristics of the reefs as well. Next slide. So we had two different methods for quantifying drusenid mussels. The first method being drop camera images. So very similar to what Matt just described. We used GoPro cameras mounted to a 1 meter by 1 meter metal frame. These frames were dropped at random across the reef complex and photos were captured. From there, the photos were scored to reflect the percent of the photo quadrat that was covered in live mussels. And each photo was scored by at least 2 people until a consensus was reached and a percent coverage was assigned to that photo. Our second method for quantifying drusenid mussels was to estimate the density of drusenid mussels on a reef using diver-acquired cobble scrapes. So with this method, divers collected a cobble from the reef and counted the number of live mussels that were scraped off of the cobble. Once those mussels were removed, X, Y, and Z measurements were collected from the cobble, and those were used for a surface area estimation. So using both the number of mussels on the cobble and the estimated surface area of that rock, we were then able to calculate the density of descended mussels on that single piece of cobble. So density measurements were collected for 5 cobbles per sample site at 6 sites across the reef. And then all densities were averaged across the site for some summary statistics. And on the next slide, we can see those. So what we're looking at here is a report of both the percent cover of descended mussels on a given reef, as well as our density estimation for those reefs. So overall, we found descended mussels on every reef that we surveyed, and the overall average cover of descended mussels on a reef was around 25%, and those numbers ranged from 8% to just above 30%. The overall average density was around 2,000 mussels per square meter, and those numbers ranged from 400 to just above 4,500 mussels per square meter. So there are a couple things to note at an individual reef scale. The first being that the percent cover and density measures were only weakly correlated. So if you look at a reef like Saugatuck, you can see that the percent cover that we're reporting here is relatively high compared to the density estimate. And then the inverse is true for a reef like Tucker's Point, where the percent cover is relatively low when compared to the density estimate for that reef. So at these sites, density measures from cobble scrapes may be more accurately reflecting the true abundance of mussels, as the photos are only able to capture the visible mussels from an aerial point of view. So it's not going to capture any of the mussels that would be on the underside of a piece of cobble or any mussels that would be covered by vegetation. That being said, cobble counts could overestimate densities at reefs where scattered cobbles are present on soft substrates or substrates that are just less likely to have mussels attached to them. Using this method, we are assuming a uniform rocky substrate across the reef. So if there are these large patches of soft substrates, then that's how the overestimation could come to be. So I think that this suggests that both percent cover and density measures are important to most accurately characterize the descended abundance on the reefs. Now, at a landscape scale, we are seeing some apparent patterns with descended presence. So our sites on the west arm of Grand Traverse Bay and the North Shore have generally high mussel abundance, whereas sites on the east arm of Grand Traverse Bay and Little Traverse Bay generally have low density and cover of descended mussels. We suspect that this may be driven by turbulent exposure at these reefs, and that in general, reefs protected from ice and wave scour will support larger numbers of mussels. So we've started to look at this, and you can see those results on the next slide. So like I said, we're still exploring this, but with some limited data mining, it seems to support this expected trend where cover and density measures are weakly correlated with depth and exposure. But like I said, we've just started looking at this. Next slide, please. So moving on to our methods for quantifying gobies on these reefs. So we calculated a relative abundance of round goby, and that number was derived from baited photo quadrats that were used on the reefs. So these photo quadrats were placed on the reef for 20 minutes, capturing an image every minute. And then later on, those images were scored where a person counted the number of gobies in each photo. And the photo that had the highest number of gobies in it was used for our relative abundance calculation. So you can go to the next slide, please. So we're looking at some of the results from that. So we're reporting the relative abundance, which is going to be the number of gobies per square meter. So we completed these surveys at 19 different reefs, and there was some variation on goby presence. The lowest relative abundance was found at Mud Lake with 19 gobies per meter squared, and our highest was found at a reef called Szechuan on the North Shore, and that was about 122 gobies per square meter. The goby abundance was generally higher at our northern shore sites, with the average abundance being 72 gobies per meter squared. So compared to sites like Grand Traverse Bay, Little Traverse Bay, or southern Lake Michigan sites, that number is almost double their average. But gobies were widespread and they were definitely found on every reef that we surveyed. All right, next slide. All right, so then finally, I'm going to discuss the methods for quantifying crayfish on our reefs. So the density of native and non-native crayfish was measured from diver-based sampling with 5 randomly placed 1 meter squared quadrats at each sample location. For each sampling event, divers searched up to 3 layers of substrate and collected any crayfish observed and placed them in a bag to be counted at the surface. Crayfish that escaped the quadrat during sampling were tallied and included in diver counts. I should note that both Virilis and Propincus crayfish were observed during sampling, but you're only going to see a report of the density as a combined native crayfish metric averaged from all the quadrats at a reef site. So those two will not be separated from each other. Next slide, please. So looking at the results from our crayfish survey, so we're going to be reporting densities here. So native crayfish were observed at 13 of the 20 reefs that we surveyed. And at more than half of the surveyed reefs, the density of native crayfish was greater than the density of the non-native rusty crayfish. Native crayfish densities were higher on core reef habitat compared to the adjacent habitat for about 10 of the 13 reefs where native species were present. The North Shore reefs supported native crayfish exclusively, so we did not see any non-native rusty crayfish there. However, we did see rusty crayfish at 12 of our 20 reefs, and we saw as many as 33 rusty crayfish in a single quadrat at our Ingalls Point Reef in the west arm of Grand Traverse Bay. The density of rusty crayfish was greater than the density of native crayfish at 40% of the reefs that we surveyed. All right, next slide, please. So some AIS management implications from these surveys. So according to a paper by Fitzsimons et al. In 2006, there is at least some evidence to suggest that round goby are a more effective egg predator than rusty crayfish. So because of this, we believe that to maximize the return on investment for control efforts, managers should consider prioritizing control on reefs with high goby density and low or no rusty crayfish. So some really good examples of reefs from this presentation would be our North Shore reefs. We also believe that directing management to isolated habitats where recolonization is less likely to occur would be a pragmatic approach. So a good example of a reef that we studied for this type of management strategy would be Potter's Reef. And then finally, for Southern Lake Michigan reefs, rusty crayfish were absent and the density of round goby was relatively low. So this might not be a priority candidate for invasive egg predator control. However, Dreissendid mussel presence was quite high at these reefs. So these reefs would be good candidates for mussel control. All right, next slide, please. So moving on to our next objective, which was to assess the fish use at these various reefs. And we kind of had 2 different methods for this as well, but I will start with our spawning assessment. So for our spawning assessment, we used egg mats. Egg mats are just a steel metal frame that are covered in this blue furnace filter material that you can see in the picture. We deployed these egg mats from mid-October through mid-December, and that was really to target the fall spawners, so lake whitefish, lake trout, and cisco. And we deployed these mats in gangs of 5 mats set both on the core habitat and non-core habitat. And we did this at 16 different reefs. So as you can see in this illustration, the egg mats sat on the bottom on the reef, and as spawners came above them and spawned, the eggs would fall onto the egg mats and become entangled in the fibers of that furnace filter material. All right, next slide, please. So looking at some of the results of our egg deposition surveys, this will be reported to you as the average number of lake trout eggs per meter squared. So as you can see here, 12 of our 16 reefs had lake trout eggs present on it. But with Fisherman Island having the greatest egg deposition of lake trout eggs. Next slide. Next, we're looking at that same metric, but for lake whitefish. For whitefish, we found those eggs on 13 of the 16 reefs that we surveyed. with the highest egg deposition happening at those southern sites. But there was still a very good amount of egg deposition happening on the North Shore as well. All right, next slide. And then finally, this is showing you Cisco egg deposition. So obviously Elk Rapids had the highest Cisco egg deposition of all of our reefs that we surveyed. But it is important to note that we did find a few Cisco eggs on Cranberry and Potter's Reef. So this was kind of a surprise to us. So those are kind of cool data to highlight. All right, next slide. So our next method for assessing fish use of Lake Michigan reefs was to use a VPS or VEMCO positioning system acoustic telemetry array. The VPS array provided fine scale movement patterns of adult fish on reefs throughout the fall spawning window. So we surveyed the movement patterns of both lake trout and lake whitefish on 3 reefs, and we did that for up to 5 years on some of these reefs. So these 2 plots that you see here are the results of some of those efforts on Elk Rapids Reef and Ingalls Point Reef. So on this figure, the circles that you see are actually going to be indicating egg deposition, with each color representing a different year and the size of the circle representing the number of eggs that were caught in that location. And then the colored polygons are going to be the results of our acoustic telemetry arrays, with the warmer colors representing areas on the reef that fish spent most of their time, and the cooler colors representing areas that fish were present in but spent less of their time. So as you look at these 2 plots, something that kind of jumped out at us was that at the spots that we were finding the highest egg deposition, it wasn't exactly lining up with the areas on the reef that fish were spending most of their time. So I think that this highlights the importance of using both adult behaviors and egg spawning or egg deposition surveys to get a complete picture of what is happening, the fish use on this reef. So like I said, some of these egg deposition spots are not lining up. So I think that it's important to keep in mind that just because a fish spawned on an area doesn't mean that that's where that egg will stay for the entirety of its incubation period. Next slide, please. So the third reef that we had these arrays set up on was Good Harbor Reef, and this was the reef that was just right next to the Leelanau Peninsula. Something cool about this reef is that both lake trout and lake whitefish were tagged, so we can really compare their ranges on the reefs, or we can really compare their ranges on this one reef. So you might notice that lake trout have a much more broad range within this reef that when compared to lake whitefish, who seem to be staying really close to that northeastern ridge. And the egg deposition numbers seem to be following this same trend as well. But looking at these figures, it made the wheels turn a little bit, and we started to wonder if there were any lake trout and whitefish interactions happening on the reef, as they are both fall spawners. Next slide, please. And so what you can see here this is a slide that was borrowed from Daryl Hondorp's 2026 GLaDOS coordination meeting presentation. And this plot is demonstrating the lake trout and lake whitefish presence over a spawning season for 3 different years. So we were interested in studying the overlap on Good Harbor Reef by these 2 species, given that their ranges do overlap. So we plan to look more into these interactions in the future, but our first reaction would suggest that there may not be substantial interaction between these 2 species, given that their peak presence on the reef doesn't seem to overlap. More specifically, it doesn't appear that their range on the reef is overlapping during those peak spawning periods either. But like I said, we're still diving into these data and plan to look into it more in the future. Next slide, please. All right, so just a brief summary before I pass the mic off to Dave, but lake trout egg deposition was highest on the main basin on the East Coast reefs. Lake whitefish egg deposition was highest on the southern and northern reefs. Cisco were mostly found at Elk Rapids, but we did add 2 new sites that they were found at. And then we're planning to investigate lake trout and lake whitefish interactions more in the future. And that's all I got, Dave. I think you're muted.
Thank you. I knew I'd do that. We're going to wrap up just coming back to a little bit about how we can use this information in management. And we talked about at the start, fish managers have several tools, but habitat management is one really important one. With respect to reefs, we can talk about protection, habitat protection, restoration, which might include sediment control, sediment removal, or mussel removal. And then habitat enhancement might be kind of limited rock placement up to and including complete artificial reef construction. So um, we want to talk about how, not sorry, slide's not moving for me, but there we go. Look at that. So with respect to reefs here, in a specific example, Taylor talked about egg deposition data that we have. Matt talked about data on kind of substrate quality, for example. If we had reefs that had high egg deposition and really good quality, we might recommend those for protection. Low egg deposition and degraded habitat, we might look at potential restoration activities for those reefs. If we had high egg deposition but degraded habitat, we might look at some kind of combination of fish management, but also habitat protection or habitat restoration work. And then if we saw low egg deposition and good habitats, maybe that indicates that some other problem besides habitat might be an issue, and so it would lead to further kind of some further evaluation. And we have this, so this is a table from our report that came out last year, and trying to get a first cut at this was sort of qualitative measures of these different variables and what we might do in terms of recommendations. So on the left is the reefs that we looked at. And then for physical, just taking again a qualitative look at physical habitat. Matt talked about embeddedness, and we had some high embedded reefs indicated in the green and lower embeddedness indicated in red. A couple of measures of invasive species, mussels and shell hash in this case. And then native fish use, and this is lake trout and whitefish eggs. And so we can see various combinations here, similar to the last slide. But if we had uh, high embeddedness and not great native fish use, which is the top couple of lines there, we might recommend some kind of sediment evaluation or sediment removal kinds of projects. We did have some really good quality, so low, in this case, low on the bottom there, Elk Rapids, Fisherman's Island, low embeddedness, not a whole lot of invasive species, and pretty good fish use. And so those are kinds of reefs that we would recommend for protection. We're hoping to, using some of the information that Matt and Taylor talked about, hoping to make this more quantitative, take a more quantitative approach to this moving forward now. And then in addition, we're just getting started with this, but we hope to use the data to look at reefs more generally across the Great Lakes, not just the reefs that we looked at. And the project we have kind of is ideally suited to taking a multivariate approach to this. We looked at a lot of reefs. We looked at lots of different kinds of data. And you've seen today landscape data, physical and abiotic information, and the biotic data. And the data was collected using the same methods replicated across reefs. So hopefully this will allow us to use this information to classify reefs and possibly do this based on as Matt mentioned, more landscape level kinds of information and things that we can get without necessarily doing a really in-depth in the field study of every single reef in the Great Lakes. So we're just beginning to work on some of this, but but really think it shows a lot of promise. So and then ultimately explore relationships between the habitat and biology. So what triggers fish to use reefs, and what reefs are successfully produce fish. So, and some of the things that Taylor just talked about. More visually, again, putting together landscape, physical, and biotic information. Um, we're hoping to be able to kind of explain and predict how fish use these reefs and how the reefs lead to successful and healthy fish populations. So I mentioned the report, and that's available for anyone who's interested. One product that we're really excited about are these reef summaries. So Matt talked about all the reefs that we surveyed. We have a summary for every one of those reefs, which include these maps. And again, shout out to Gus Annis he did a ton of work and a really nice job putting these maps together, really useful. I think will be really useful in the future. They were great for us, but will be useful for others as well. And then in addition to the maps, we have background information about each reef, kind of including references to historical studies, all the physical habitat summary, fish use summary, and then talk about what kinds of impediments and impairments there might be on the reef to hopefully direct some of the future management. So really, really proud and excited about having these out there, and looking forward to kind of building on this effort in the future. And then just wrap up, any project like this requires a huge amount of work and is made a lot better by having lots of great collaborators. So just acknowledge everybody here that collaborated. A big chunk of the money for this came from the Great Lakes Restoration Initiative through EPA. The Nature Conservancy and Michigan DNR also provided funding. Chris and Eric at Grand Traverse Band, huge insights into, we talked about those reefs around East and West Grand Traverse Bay, and kind of how they were, some of the really interesting patterns developed there. And Chris and Eric really, really crucial to understanding some of that. And then at USGS, Pete Esselman's group, really a lot of the work we did relied on some of the data that they've collected through the work that they're doing around the Great Lakes here. So acknowledge them as well. So with that, back to Bo, and we'd be happy to answer questions if there's time, Bo.
Yes, definitely time. Thanks so much for all that information and the impressive amount of work you all compiled. I mean, my first question is, where can people find that report?
Yeah, I'll start, I guess, and we can Matt and Taylor with this one. We can you can get it from us. It's
Okay.
I don't know if Patrick's on right now, but or Taylor might know if the DNR has it posted internally at all. We don't have it. We don't have it posted unless they've done that recently.
Okay.
But definitely get it from us. So contact.
Yeah, okay. So the best way is to email you all.
Yep.
And it seems like really nice summaries, especially those reef summaries that you said. So for questions, you can pop them in the chat. Ideally, you can do what Vic is doing and just raise your hand and then pop your camera on so it can be a little bit more personalized. But yeah, Vic, why don't you start us off?
Oop,
Here
I
you go.
think
We
I'm
hear
good
you.
now. Can you hear me
Yep.
now? Yeah.
Yes.
No, great work, guys. This is really a cool study. I've been kind of following along over the years that it's kind of been going on, and it's great to see the results. And I really like those summaries and the way you guys, like you said, Dave, you got landscape, physical information, and then the biotic information to boot, covering everything that really gives us information on how these reefs are, productive for fish populations and stuff. So that's awesome. I do have three questions. one of them on the telemetry part of it. How many receivers? Maybe this is for Taylor, but I'm curious how many receivers it took around some of these reefs to get those kind of heat map kind of distributions and things.
That's a good question. I don't actually remember what the exact number was. I want to say somewhere in the realm of 30 on on Good Harbor Reef.
Okay,
But it was quite a few of receivers to do this fine scale array.
right. Okay, that's what I thought. I was just kind of curious. And then maybe back to you, Taylor, or one of the other guys. You had pretty cool density data, I thought, for gobies, and I wonder how that relates to some of Esselman's work, if you've had a chance to look at that as far as his estimations of goby abundance. I don't know if he ran transects up in that part of the lake, but I'm just kind of curious if you've taken a look at that and if they're pretty similar.
We haven't directly compared. I don't know, Taylor, jump into, but we haven't directly compared numbers by site with Pete's numbers. We've talked about doing that, just haven't.
Sure.
It's a great
Well,
question, though. Like, it'd
Yeah,
be really interesting if Pete sees higher density along that North Shore as well.
yeah.
yeah, I just wonder if the trends were the same. That would be kind of lend validity or whatever to both of the methods, maybe. And then I don't know if you'll be able to answer this, but a lot of our lake trout spawning sites are at our offshore reefs, although I think we may be getting spawning near shore as well. We just don't monitor that as much. Uh, how deep? I mean, it seems like most of your reefs up there are pretty shallow, or the ones you looked at. And any recommendations on, getting similar information from some of the offshore reefs, stuff that's, 90 to 120 feet in depth.
Yeah, I think a lot of these methods could definitely be used for that. We did not. We wanted to hit as many reefs as we could, so we decided like we're just going to focus more on more nearshore reefs. They're not all near shore, but like
Yeah,
Taylor talked about, Potter's Reef was, surrounded by deep water, but most of them are nearshore reefs that we could access pretty quickly. We're worried about, you know, having egg mats out at a ton of reefs, and that's a lot of work to keep processing those. And so we wanted to hit as many reefs as we could, and we decided to focus more near shore. But I don't see any reason why uh, just about everything we did couldn't be done on the reefs you're talking about, Vic.
okay. Well, really cool study. Appreciate it. Thanks for your questions, answering the questions too.
Yeah, thanks for the questions, Vic. While we wait for another question, yeah, Corey's up. I was just going to ask about the differences between the east and west arm. I mean, it seems like Elk Rapids and I think Birch Lake were just really beautiful, clean reefs. And does it have to do with depth, or what do you all think is really driving the differences between the quality of the reefs in those nearby but in very different arms of Grand Traverse Bay?
We think it's exposure. That east arm is exposed to the northwest winds, which is a lot of those fall winds that really shape the clear out sediment and stuff. So we think that exposure really makes those reefs, what they are. Obviously the the geology there, the natural geology too. But then along the west side, there's certainly a lot of rocky base to those areas, but there's just so much silt that's built up, and then definitely a lot of drysinids that are covering those reefs. And I think the drysinids and the sediment almost kind of interact with each other. The more drycinid buildup you have, the harder it is for wave action to work its way into there and clean them. And there just isn't very much wave action on that side to start with. So you just end up like really building up material in those spaces.
So that cool relationship you're beginning to see with like depth and percent cover of mussels, I guess. So that looked like the shallower reefs would potentially be more vulnerable to that high wave energy. So is it maybe some interaction between like the direction where the prevailing currents are coming plus the depth?
I don't those particular the east versus the west arm. I don't think so because those west arm reefs are pretty shallow too. I think something like Good Harbor, which has a lot of Dreissenid buildup, I think that is a depth. Yeah, we definitely see that too. We see some of those deeper sites having more either more Dreissenids or more sediment, and that's I think a function of the depth. But the west versus the east arm, Taylor, Dave, correct me if you think I'm wrong, but I don't think that has to do with depth. I think that's more just exposure. But you're certainly right that there are other reefs that are deeper that we were seeing kind of the same pattern.
Hmm.
The
Yeah.
other thing we didn't really talk about, Matt, but those east reefs, it's really interesting how well like the rocks are sorted compared to some other reefs, and I think that points to more of that exposure
Hmm.
exposure kind of factor too. But they're just really beautiful, kind of uniform rock formations down there, and so
Nice. Let's go to Corey, and then after that, we'll get Ben Leonard's question in the chat there.
Yeah, thanks. Matt, Dave, Taylor, that was an awesome update. I'm so excited about all the spatial things I saw. I too have a spatial question, kind of like Bo. I was really intrigued by lake whitefish using the northern and southern reefs there, and was wondering if you all had any hunches on what's going on there in those middle regions, especially given Gobies seemed pretty dense up north. Like any hunches about that spatial question, the split with north and south being high use?
there's
Stumped.
Taylor, I can speak a little to it, I guess. There's some in those fact sheets. We didn't have adult data. We didn't go out and gill net every week on all these reefs, just too much effort. But we did put information there about kind of stock size in those areas, and I think the stock size is a little bit higher of adults in those areas than in some of the other areas, which might explain some of that. But uh, the other thing down south, at least, this is really more my opinion, I guess, than anything. But it's kind of a limited. Like up north, you have reefs all over the place, kind of, and down south, you have a little bit more kind of isolated reefs. So possibly the fish populations that are down there are kind of crowded into one area. And again, just maybe a hunch or an opinion, but I think the Saugatuck and that Ganges, we really saw a lot of egg deposition, and there's certainly not the reefs down there that there are up north. So
I think
Oh, go ahead. Yeah.
I think that point Dave just made, I think that's really probably the primary reason. I'd like to think that maybe, those reefs down by Saugatuck, that's really close to the Kalamazoo outlet. The St. Joe outlet is not too far south of there. I'd like to think that maybe there's more nutrients down there, but I'm not, to help with things, but I'm not sure other data sets are really supporting that. So it's probably not related to that, but.
Yeah. It'd be interesting, like, to look at the distance to shoreline or nursery areas from these and see if there's a connection with southern and northern regions too. I mean, I think it'd be interesting to look at. I'm just thinking of spatial questions and also larval densities being so low down south to see, like, if those reefs are higher quality down there and if they're supporting a lot of spawning, but yet not, if we're not seeing the larvae. That would be a really cool question to look at, or if they're just going somewhere else. But yeah, good stuff. I just was curious.
And if I
where?
could add just one other thought too for the high numbers of egg deposition on the northern shoreline, I do think that, again, this is probably more of an opinion, but some people are believing that we have some influence from the Green Bay whitefish that are spawning in that area, certainly on reefs like Wiggins. I think that there was a grad student who seemed to find some evidence of Green Bay fish coming in to spawn on those reefs.
That's great. Yeah, intriguing too. Yep. Thank you.
So Ben Leonard put a question in the chat about round whitefish eggs. Did you all, if they were there, would you have been able to tell them apart? I actually don't know if they're different enough in size to be able to distinguish from a trout versus a whitefish lake versus a round. Maybe you know, Ben. Do you know?
I don't know anything about their eggs.
Okay, good question. You stumped everybody, maybe.
It is a great question.
We did use, I don't know if Taylor, did you mention that? We did use size, and then back we're analyzing the two size groups of Gorgonid eggs to make sure of what we had, um but I don't
And
know about round whitefish.
yeah, we've done genetics. There's been genetics work in the past that have kind of supported the size differences. But, here we are working on a lot of new sites. And so if there's round whitefish on some of those reefs, yeah, it is a really good question.
Yeah. Our may have that, at least the size. We could look that up pretty quickly here on the fly if anybody has their electronic copy of our at all. Um, so I had a kind of rusty crayfish question. That was interesting. Boom. There you go, Corey. So that's pretty. Can you put the lake whitefish and lake trout diameters in there too from our, Corey? We can see if there's overlap. And Cisco, I guess. Um, So the rusty crayfish stuff, that was looked like they were like concentrated in maybe two regions. Is that because they are still invading and expanding, or is it just because that's just potentially ideal rusty crayfish habitat?
I don't know, Dave, do you have any explanation for that?
Again, my just a gut. I think they've been around long enough and not based on some other, gobies kind of came in and peaked, after 8 years maybe or so. So I don't think I guess I wouldn't say that Rusties are still in an invading stage, but we don't I don't yeah, like I don't know what their habitat requirements are compared to Rusties. That's one of the things just finding all those areas where there were native was a really interesting finding,
Hmm.
but hopefully we can put together some of the other data and say, maybe these are habitats that are less prone to invasion or
Right.
more suitable, but we don't really know right now, I don't think.
Okay.
Yeah, ideally there would have been surveys like this 20 years ago so we could compare against, but
Hmm. Yeah. Yeah. Go ahead, Vic.
Yeah, I was just wondering, this kind of management, based on your management recommendations, you have some really quality reefs and you talked about protections for those reefs. I'm just curious what you had in mind in terms of what the protection would look like. We've done stuff with commercial fishing and with lake trout restoration. We have no commercial fishing on reefs, sometimes no commercial and recreational fishing on some of the refuges. What are you thinking on some of these reefs? I'm just curious what it might look
I
like.
th I think, and jump in, Matt and Taylor, but I think mainly right now what we're looking at is more physical protection, and that's some of those landscape variables. So like dredging activities that would be nearby that might increase sedimentation rates on those reefs, or we didn't talk a lot about fisheries management activities, like specific, right, regulations or but it definitely could be part of it too. But I think we were more focused right now on kind of physical habitat effects and what that landscape analysis tells us. So
Yeah, so really just identifying them as high quality areas to be protected. I mean, maybe there's some kind of category or something that could be developed or, where you could had a way to identify them as protected areas. I don't know what that would look like. I know we do that kind of thing on land, you know, with natural areas and that kind of thing. It'd be kind of cool to see something like that maybe in these really important fish productivity areas. It's not just fish too, invertebrates, everything. So
yeah.
yeah, cool. Thanks.
Yeah, I think at minimum, just getting information on the best quality reefs in the hands of, regulatory agencies would be really valuable, I think, just so when harbor expansions or dredging projects are being reviewed, like it's really handy and mindful that there's some really high quality habitat there.
Yeah. And I don't know if it's ever going to happen, but offshore wind and that kind of thing.
Yes.
It's really important, I think, to have these as well as fishing locations and that type of stuff identified. But thanks.
Thanks, Vic. Good question. So Corey has been putting the diameters down there. It looks like there could be some overlap between rounds and lake whitefish, but um, yeah, there definitely is a genetic test for that. I guess if you saw a larger than expected lake whitefish egg, it's possibly that could be a round. Um, Here for any of the questions. So wasn't there some larval fish work done on a subset of these reefs? Maybe you could just talk a little bit about, maybe you don't have the data summarized yet, but was there anything that sort of was interesting that came out for some of that larval fish data that didn't show up for some of the other fish sampling data?
I mean to go. It's kind of I guess I would say it didn't really correlate well with the eggs, and they're tough to sample. So we were doing larval tows where we could, and we were talking about this actually yesterday, that
Hmm.
it's hard to attribute larvae to a reef, and we didn't
Drift.
just because they're drifting around, and we did not see like places where we saw tons of eggs. We didn't necessarily see tons of larvae. So um, we're still trying to put some of that together, but it's that's a problem, I guess, for these studies. It's kind of I know Taylor's been working with traps with emergence traps right now, and they're probably, if they work, they tell you more about what's going on in the reef, but they don't seem to catch a ton of fish either. So
Yep.
don't have a great answer for you, Bo.
No, no, that's great. I mean, because I'm thinking about that in terms of what would you call them, sort of the engines of the Great Lakes or something like that. And are there ways that we could be monitoring them in ways that are like we know it's coming from the reef, right? So I'm thinking about zooplankton or primary production, thinking about like some of the work that's like the lake mount idea that was recently sort of proposed, how almost like creating these productivity hotspots as the currents are being forced up and bringing nutrients up and creating productivity. But it would be the same issue because of the currents that quickly move the plankton, whether they're ichthyoplankton or zooplankton, away from the reef. So it is a challenge. Mhm.
Yeah, if you look at in those fact sheets that we produced in the report, they're more the fish data other than the eggs are more of a regional kind of summary, not a reef specific summary, just
Mhm.
because it's hard to attribute that stuff. So
Yeah. Well, 42 people are still hanging on. Um, any final questions from anybody? I could ask kind of a fun one because you all are reef experts and I'm not. Like, what other fish species do you think are spawning on the reefs at times a year you didn't sample?
Funny enough, Bo, we just got a bunch of walleye larval fish on Big Bay Denok, so certainly
Okay.
walleye.
Okay.
we'd
Well,
see
that's
rock bass and smallmouth bass, during summer sampling or spring summer sampling on a lot of these.
mhm.
Probably perch.
Yeah. Okay. All right. Well, I think we will call it right there. Thanks so much for pulling together a really informative and helpful presentation. Reach out to Taylor or Dave or Matt via email. And if you don't have their emails, you could reach out to Renee or me, and we could put you in touch with them if you'd like to get a copy of that great report. But at some point, I assume it'll be out on maybe a Michigan DNR state report library where there's often a lot of really good resources there. So a lot of virtual hand claps there for you all. And so thanks again. And we look forward to seeing you all in early June with Scott Koenig-Bauer's work talking about Cisco restoration in Saginaw Bay. Until then, have a great weekend, everybody.
Thanks, Bo.